next speaker is Dr. Christopher Lucas from the University of Edinburgh, and the title of his talk is SARS-CoV-2 Pathology in Humans.
Thanks very much to the organisers for the invitation. It's really a pleasure to have this opportunity to share some of the results from our ICECAP COVID-19 post-mortem study. So thanks very much. So I thought I'd start initially by just actually giving a disclaimer to say that I'm neither a virologist nor a pathologist. So be kind when you're asking me any questions. I am, however, an adult human physician. And in February 2020, I'd just relocated back from the US to Edinburgh to set up my laboratory in the Institute for Regeneration and Repair, which turned out to be quite a challenging time because within a month, the university was essentially closed. And I was back in clinic almost full-time, looking after this enormous wave of huge numbers of patients that had hypoxic respiratory failure. Large numbers of them were dying due to this novel coronavirus. And we knew quite early on, unfortunately, that this was something that was going to unfortunately kill quite a lot of humans. The purple bars in the middle are deaths, and we knew quite early that this was a disease that was primarily going to kill older people and with a male preponderance, which obviously was quite alarming to an awful lot of the academic community. Now, with respiratory disease, and particularly in the context of pathogens and viral pathogens, we know that inflammation, organ injury, and death can occur either due to direct viral toxicity, it can occur from inflammation from an appropriate response secondary to the pathogen, and it can occur due to aberrant, exuberant inflammation that was initially precipitated by the pathogen. And I think it's really useful to think about this because I think it informs how we think about potential approaches to therapy and whether we think that resistance, where we target the pathogen, is a good approach, or whether we think about tolerance, where we aim to prevent injury despite the pathogen presence. So what we wanted to do was to set up a multi-parameter tissue survey in humans, really to try and answer what were the key features of fatal COVID-19, try and understand if the disease was due to direct viral cytotoxicity or dysregulated inflammation, and also to understand a bit about the immunological consequences of infection at a tissue level. Because in lots of organs, and particularly the lung, the only real way to understand what is going on is to actually get a bit of whole tissue. So surely all we had to do was just to tap into the existing UK pathology, autopsy and post-mortem network to make all of this a reality. Now, unfortunately, post-mortems in the UK have a bit of a bad image. There's been a few high-profile scandals, largely related to sampling and retention of tissue and organs without the explicit consent of relatives. And partly because of this, and also because of changing practices and thoughts amongst clinicians, the hospital post-mortem rate has absolutely plummeted in the last few decades. So in the UK, less than 1% of all patients that die in hospital currently get a post-mortem. However, the potential utility of this approach was picked up even by the media, and this again was independent as well And I think the Independent said that only a few dozen post-mortems had been carried out by this early stage, and actually I think we'd carried out over half of them when this article went into press. So essentially we had to set all of this up de novo, which was probably the most difficult thing about the whole study. So we enrolled patients that had a pre-mortem diagnosis of SARS-CoV-2 infection and either lung injury or myocarditis. We had a referral hotline that clinicians from any of the 3 major hospital sites in our region could refer to. We had 2 really fantastic research nurses that would go out and take consent from relatives to both perform a post-mortem examination and also to take tissue for research purposes. And then what we did after that was something that we called a warm post-mortem examination in our high-risk category 3 autopsy suite. Now, why and how is this horrible phrase of a warm post-mortem examination relevant? And it's really because after death, tissue cytolysis sets in and you immediately start to lose lots of really useful information. So in the top 2 slides, you can see this in the kidney, that after 4 hours after death, you can see the tubules, you can see individual cells, and you get a high degree of granularity. However, after 50 hours, a lot of this information is lost. So we aimed to do as many of our postmortems as we could within 24 hours. And how we did this was we recruited David Dorward, who's our thoracic pathology lead in our region, and David actually did every single one of our post-mortem examinations and a huge amount of the downstream analysis afterwards. And David was co-lead on this study. So in terms of who we recruited, I'm not going to dwell on the demographics too much, but essentially we recruited people that were generally older, predominantly male, comorbid, and about a third had been mechanically ventilated. So actually very typical of the typical patient that died from COVID-19. And this was all done very early in the pandemic, so this was all pre-vaccination and pre-routine treatments, although a couple of our patients did get experimental medicines at that time that are now part of the routine treatment in the UK and elsewhere. So what we did was a standardized post-mortem examination where we sampled from 37 distinct tissue sites to really get a really broad overview of what was happening. Over half of the tissue sampling was from the lung and respiratory tract because we knew already that was where most of the action was likely to be. And the first question we had was really, well, where is the virus? So given I'm not a virologist, we set up a really fantastic collaboration with Julian in his lab, who led all the virology work for this study. And what the little green human here shows is essentially all the different tissues that we sampled, with the increasing intensity of green showing the increasing frequency of viral RNA presence at all these tissue sites. And what you can see is that, yes, the lungs are very dark green, as you would expect, but essentially this virus has the potential to be in all sorts of tissues all over the body. And every single tissue that we sampled, we had at least one patient that had detectable virus at that site. And when we split this out on a per patient basis and also stratified them in terms of illness duration, again, what you can see is that there's really huge variability. So that the first patient that unfortunately died under 10 days from illness onset has virus in pretty much every tissue. Patients 3 and 4 essentially have virus in a couple of tissues. And then the 2 patients that died over a month after disease onset have virus in lots and lots of tissues. So essentially this can be everywhere and persist for a long time. So certainly lots of parallels with the previous talk. And when we looked for a correlation as to whether there was a correlation from illness onset to the number of virus tissues, there was essentially no correlation whatsoever. So all of this was done with multiplex PCR, and Julian and his team confirmed a lot of this with viral sequencing, with reads mapping to the SARS-CoV-2 genome. And although we weren't set up at that time to do viral culture, the sequencing also revealed evidence of subgenomic mRNA consistent with viral replication at many of these tissue sites. So, having described where the virus is in all these tissues, surely when we looked at inflammation, it would essentially be exactly the same as where the virus is, with the thinking that if organ injury is primarily collateral damage to the virus or an appropriate viral-mediated inflammation, there should be temporal and spatial match with inflammation and viral presence. And essentially we found the complete inverse of this, where essentially the lung and the reticular endothelial systems, so the spleen, lymph nodes, and bone marrow, were really the exclusive sites of inflammation in these humans. And most extrapulmonary sites, even with detectable virus, had absolutely no evidence of tissue inflammation. So to try and drill down into this in a bit more detail and actually to understand on a cellular basis where the virus was, we performed SARS-CoV-2 protein detection using immunohistochemistry. So in these tissue sections, the blue dots are cell nuclei and the brown dots are viral protein. And what we can see is that in the nasopharynx and the bronchus, so the upper airway, as well as the gut and the liver, the S protein is pretty much exclusively within epithelial cells. Of interest in many extrapulmonary sites, such as in the liver, we very frequently observe these runs of viral positive cells, many of them all sitting together, often directly adjacent to lots of cells that were negative for virus, which to us suggested the possibility of direct cell-to-cell viral spread that I think has been described for avian coronaviruses in the past. And what about in the lung? Well, this was essentially the same sort of thing, but with lots of different colors. So here we have yellow epithelium, green endothelium and pink macrophages, and the S protein in red. And essentially what we found was that the vast majority of the virus was in the epithelium, but there was also some present within the endothelium and also within the macrophages. However, in contrast to the non-pulmonary sites, at the level of the alveolus, which is right in the distal lung, we found that the viral S protein was very patchy and spread around, perhaps consistent with spread from the upper respiratory tract. So this actually looked quite different to what we saw in some of the extrapulmonary sites. So that's in terms of the virus, but what about in terms of inflammation? Just to orientate you, this is what a normal bit of lung looks like at the level of the alveolus, where gas exchange happens, and essentially it's lots of white space with nothing there because that is the purpose of the lung. And very commonly in critically ill patients, they can develop this clinical condition called ARDS, which is acute respiratory distress syndrome. And the pathological hallmark of this is something called diffuse alveolar damage, which is essentially really substantial epithelial injury at the level of the alveolus. So did we observe any of this, and did we observe anything else? Well, the top two pictures are actually still lung, even though this doesn't really look like lung at all. It just looks like a solid bit of tissue. And this is what DAD, so the pathological hallmark of ARDS, looks like. In addition, we saw some other things. So we saw some pulmonary thrombus, we saw some bronchopneumonia, so full of neutrophils for the neutrophil lovers in the room. We also saw patients that had essentially normal lung, and we saw patients that had normal lung directly adjacent to wildly abnormal lung. So a really, really heterogeneous mix of findings. And when we split this up on a per patient basis and on a per lung lobe basis, we scored this all just in a binary fashion in terms of presence of viral RNA in green, presence of diffuse alveolar damage in orange, thrombosis in purple, and bronchopneumonia in red. And hopefully just a quick look at the colours can convince you that actually different patients were very, very different. And just to highlight three in particular, so patient F that's at 6 o'clock position, there was virus in every lobe of the lung, and there was diffuse alveolar damage in every lobe of the lung, so this kind of made sense. However, in patient I, that's about 9 o'clock, there was virus present in every lobe of the lung, but no diffuse alveolar damage. And in patient J, there was no virus, but lung injury. So lots of things that actually didn't quite match up. And it was really this mismatch between the viral presence and inflammation and organ injury, both between the organs globally, but also within the lung tissue itself, that led us to conclude that actually immunopathology and not direct viral cytotoxicity is a primary mechanism that can drive disease severity during COVID-19 in humans. So now I'm just going to very quickly focus on about 3 different pathological findings. So there's quite a lot of interest in blood clots. So we saw blood clots in actually quite a lot of our patients. So 8 out of the first 11 that we examined. These were present in both large and small vessels. And while blood clots can and do kill people directly, none of these looked substantially enough to have been the direct cause of death. Some of these were diagnosed pre-mortem, but actually the vast majority had not been diagnosed pre-mortem. So this actually very radically changed how we looked after and investigated our patients in the clinical arena. And just as a comparator to other diseases, just to try and give a flavour as to what might be truly SARS-CoV-2 specific, while increased thrombus has previously been reported in SARS, flu A, and ARDS more generally, it seems in COVID-19 the incidence of thrombus was about a log order higher than that previously found in flu A. So while the presence of thrombus may not be truly COVID specific, the incidence of it certainly seemed to be. In addition, we also saw pulmonary vasculitis, so it's nice to hear that vasculitis has been reported in other contexts with coronaviruses. So this image here, the slightly pink circular structure in the middle is a blood vessel in the lung, and the little purple dots are nuclei of inflammatory cells that absolutely should not be there. And when we immunophenotyped these inflammatory cells, we found that these were predominantly monocytes and macrophages with smaller numbers of T cells. And interestingly, we didn't find any evidence of SARS-CoV-2 protein within these blood vessels, and we didn't see any vasculitis in any non-pulmonary organs. And with the look to previous literature, while perivascular lymphocytes have been described in flu infection, these are essentially lymphocytes around the blood vessels rather than in, so this may well be a very specific COVID finding. And when we phenotyped the whole lung itself, so these very solid-looking abnormal bits of lung, what we found is that the inflammation was predominantly parenchymal as opposed to vascular. And again, this was mononuclear phagocyte predominant inflammation with huge numbers of macrophages and monocytes in the lung with much smaller increases in T and B cells. And actually, what we now know is that there can be altered monocyte macrophage numbers in many different causes of lung injury. So this paper that came out during the pandemic essentially showed that in most causes of lung injury, whether it's flu, ARDS, bronchopneumonia, or COVID, generally you get depletion of the alveolar macrophages, which are the cells that live within the lumen of the lung, but there's always this very substantial expansion in what we call interstitial macrophages that live embedded within the lung tissue. Now, what we don't yet know is whether these interstitial macrophages have antiviral roles, whether they're involved in tissue repair, or whether they're just being recruited as part of uncontrolled inflammation. But this is something that my lab is taking forward and investigating at the moment, particularly the role of these cells in tissue repair and regeneration. So from everything that I've shown you so far, can we actually make any conclusions, and are there any caveats? Well, maybe the caveats first of all. So we didn't recruit any non-COVID cases into our series because this was all done as an emergency response to the pandemic. And we mainly compared to normal lung and the literature, particularly using flu A as a comparator. We had a fairly heterogeneous patient group, and I think probably the major caveat for any post-mortem study is that, of course, we could not and never will be able to recruit those patients that got severe disease and then recovered and survived. And I think that actually that is always going to be a limitation of this sort of study. In terms of conclusions, well, essentially what I've told you already, that this lack of temporal and spatial association with inflammation and virus led us to conclude that immune-mediated organ inflammation, so immunopathology, was driving severity. And this is certainly consistent with the findings that immunosuppression can prevent death in severe cases. So does this mean that we should just give immune targeting therapy to all patients and forget about everything else? Well, absolutely not. And I think the reality is actually much more nuanced than that. And the results from our study that certainly support that statement is from a proteomic study where we investigated lung and spleen, so the two tissue sites with extensive inflammation. And what we did is compared this to normal lung and essentially found that our normal lung and our COVID cases separated quite nicely. And like any good omics study, some stuff was up and some stuff was down, although I'm not going to dwell on any of the individual proteins other than to say that lots of them made logical sense. But what we thought was actually really quite interesting from the study is that when we did some unsupervised clustering, so this is when you just ask the computer to find patterns in your data and you don't tell it what the data actually are, it separated our cases into 3 different clusters. The one at the bottom contains every single one of our control normal lungs, and the top 2 contained the two clusters of COVID patients. So we wondered what might be different about these 2 clusters of COVID patients. And when we inspected the metadata, which for clarity was not involved in the clustering algorithm, we found that 3 things were different. And that was illness duration, presence of viral RNA, and the inflammation severity. So the patients that had a short illness duration generally all had viral present, and they generally had mild to moderate inflammation, whereas those patients that died later generally didn't have virus, but they all had moderate to severe inflammation. So we termed these 2 clusters early viral and late inflammatory clusters. And I think what this tells us is that fatal COVID is not just a single disease entity, and that there's very clearly different disease stages, and that gives us a real opportunity to think about how we might target this therapeutically. And certainly if you have a patient, whether that be a human or a cat, it may well vary whether you're in the early viral or the late inflammatory stage, how we think about treatments for this. So I'm going to finish there because I know that I'm standing between everyone and lunch, just by thanking everyone that was involved in this study. Particular thanks to David Dorward, who's our pathology lead, and Clark Russell, who is our infectious medicine lead, as well as Kenny Bailey and David Harrison, who were the other investigators on this work. Huge thanks to Julian and his team for doing all the virology work in what was still our most fun COVID collaboration, and of course the huge numbers of clinical, pathological and laboratory colleagues that contributed to the analysis of tissue and data. And we got some funding, but a particular thanks to all the families that very generously consented to enrolment in our study. And if there is time, I'm very happy to answer any questions either now or over lunch, including, yes, that is a blue sky in Scotland. So there you go.
Any questions?
Yeah, because this all derailed it. Yeah.
Test. Excellent. So we did minimal invasive tissue sampling because the mentality there is against full post-mortem. And what we found, there was huge variation within the lungs. So some areas might have diffuse alveolar damage, some others may have way more healing, some others may have way more thrombus. It's something you have seen in our lungs, we're more like a mosaic, rather than all of the lungs have the same difference, the same pathological process. It's something you have seen. How you explain that?
Yeah, absolutely. So as well as a huge variety in the sorts of pathology that we saw, very frequently saw really, really highly abnormal areas of lung directly adjacent to lung that actually looked like it was pretty normal, which I think tells us that, you know, there can be different processes going on. Yeah, there's virus, the immune system can be very different, and I think we now know that actually a major signal to recruit monocytes and macrophages in the lung is epithelial injury, and of course a lot of these viruses target epithelial tissues as primary drivers. And that's a slightly hand-wavy answer as to why things can really vary so substantially, but that's also something that we observed in quite a number of cases. Yeah.
Hi. Yeah, lots of good talks, and thank you for that. I just wondered, you talked about looking at transcriptomics and the omics parts.
Mm-hmm.
Were you able to look at whether or not there were any genetic differences between those two groups that you described at the end?
Yeah, so we didn't look at the sort of human risk studies, but actually one of our proteins that was differentially regulated was MCP3, which is a ligand for CCR2, which Kenny's team showed was a major risk factor for COVID severity. So certainly within that data set, there was actually several proteins that mechanistically made sense, including ones that would link with genetic severity.
But those aren't being considered biomarkers for different ways of treating.
Yeah, absolutely. I mean, like MCP3 would be a really good example of that. You know, we also found other things such as NRAGE, which is already a pretty well-established biomarker of lung injury, was also differentially regulated between these groups.
If I understand rightly, we're not actually genetically testing people when they come into those situations, are we?
No, we're not.
Okay, thank you.
I'll leave the last question for me. With experience in the hepatitis field, we saw that people progressing to chronicity or responding to antiviral treatment, that was very much dependent on HLA types. Mm-hmm. This is something that's not been investigated extensively with COVID or with the feline, because presentation of antigens plays a major role in the immune response. So any idea whether...
Yeah, so that's not something that we looked at. I think just to maybe say to this audience is that we did obviously take a huge amount of tissue and we set some of that up as a biobank, so there is actually lots of tissue still available that maybe some of these questions such as MHC expression and some of the genetic studies could actually still be done in these tissue sections that we collected.
Yeah, and one other example of that is CMV, which downregulates beta-2 microglobulin expression, you know, and viruses tend to do that in an attempt to become chronic.
Yeah, so I don't think we directly measure
That's
beta-2
generated more questions.
microglobulin.
I'll just shout, wonderful work, really respect it. Your comment about the decrease of postmortems, has any, I'm going to say qualitative, I was going to say soft, but it's not the right term, qualitative work been done to say, look, this is wonderful work that could only have been done through the generosity of families doing postmortems?
Yeah, so we have a draft paper where we're trying to address that, because I think at the start of this study our biggest concern was actually, because of the circumstances of the pandemics, lots of these relatives hadn't actually seen their family member that had died for 2 or 3 weeks. They'd been kept out of hospital. Everything was done over screens. We thought that actually approaching them might be a bit of a nightmare at a really kind of critical and sensitive stage. So we actually collected a whole bunch of feedback from their relatives post-event, and it was unanimously positive in support of the way that we'd handled it, and also the fact that their relative, had potentially contributed to something actually useful, even in the most awful of circumstances. So I think certainly locally, I'm very vocal as a clinician about the utility of post-mortem examinations, but I agree that I think that's probably a question that we should try and push and revisit on a slightly more global scale. Great.
Okay.
Great. Thank you.
Thank you very much.
Thank you.